I'm going to present our system called Persephone. It's basically it's a powerful genome browser. It's a system for storing and visualizing genomic data with some analytical tools. We've been working on it for more than 10 years, and now I wanted to present the web version that will run in your web browser and is more portable than the Windows version, the original one. Okay. You can start it right away. Just go to this website, web.persephonesoft.com, open the page, and there we go. So it will automatically start the web application that will show you a few public genomes. One of them is Arabidopsis. We have human data, virus data, wheat, and so on. Okay, so Arabidopsis. This what we call map set or genome, which contains a set of maps. Yes. I open the first chromosome of Arabidopsis. As a traditional genome browser, it shows several tracks of data sequence with as a GC content and or polyns, gene models, marker tracks, quantitative tracks, and typically it's RNA-seq coverage, a few others, and BAM data, BAM files. You can upload your own data, and you see this blue background is actually it's telling us this is a data, my own data. So it's visible to me only because I just uploaded it. So what you can import on top of the data from the database is markers, BAM files, markers in the format BAM files. Faster, you can drag your own genomes, so like full genome if it fits the quota, or if it doesn't fit, talk to us, and GFF file with annotation. Okay. If you want to see more details, so you see this is the entire chromosome, and you can zoom in by just rolling mouse wheel. So you need the mouse wheel, yes. Or you can just with a shift, you select the region, it will zoom in, and what's important, it's smooth. So the animation is smooth, so you will never lose your focus, unlike other genome browsers, right? So you just zoom in and you see the sequence and more details on the fly. Okay. If you want to see more splice variants, you can expand the strand, and now you will see this in more details. Let's hide it again. If you click a gene, you will see its properties, basic properties or qualifiers in the form of key value pairs. Some of them can be converted into hyperlinks to external sites. Protein sequence, splice sequence, translation, which is spliced with overlaid translation, or a splice sequence, which is just a color coded sequence with portions like exon, introns, and so on. And also a metrics tab which tells you the sizes, distances, and so on. Okay, I have to jump through all this because I have just 15 minutes, but we will just cover the names of features, not the features themselves, sorry. Okay, so for the BAM file, as long as we're here, we can just zoom into that. And we can see the individual reads, like exon boundary, and I can expand it actually, and I will see individual reads with some SNPs. I can zoom further and I will see individual nucleotides if needed, but sometimes it's too much, so I will just hide non-variants, so it's much clearer. Okay. Persephone is not just a genome browser, it's a multi-genome browser, so it allows to align maps and align maps of different kind. For example, you can take a genetic map and physical map based on sequence. This is sorghum. Okay, so this track with markers in genetic map, you see this brown color, it's a genetic, and this is based on sequence. Same markers or subset of those appear on both sides. Okay, if it's the same marker, it automatically linked by this thin line. And sometimes it's important to compare genetic map to sequence map because it can reveal some issues with assembly. What's also interesting is that once you click such marker, it will give you, first of all, it will give you the sequence that was used for mapping this marker. So this sequence helped to map this marker on sequence map. But also it shows all locations for the marker. So you know now this marker is been mapped on some location in centimorgans in genetic map and also in some sequence. One of the most popular features of Persephone is its BLAST interface. You can call it from here, provide your query sequence and select your subject and run whatever BLAST program, or if you already have a protein or gene of interest, this is rice, you jump to protein tab and take its sequence by clicking this button, so it will just copy the sequence into the query box. And my task is just to find orthologous gene in sorghum. So I will select sorghum entire genome because I don't know which map contains my ortholog, and I can tweak the parameters and run t+n, where my query is a protein and the subject is DNA. It takes a few seconds and this is the result. It's presented in the form of this data grid where each line corresponds to an HSP, and HSP is high scoring pair from BLAST output, and you can just navigate through this by clicking. So we found that our ortholog is somewhere on chromosome 3 close to the end. So let's take it and just display map and close. So now we're looking at the sorghum chromosome 3, which is aligned to our query here. So this is a track that we just created. It contains BLAST hits, and you will immediately notice that the HSPs are consistent with the gene that was predicted in the same region. But sometimes the HSPs are much longer than exons. What's going on? So this may confuse your algorithm if it takes into account the protein hits. So if you click this one and look at the alignment, you will see, aha, something is going on. So you have this hit, but it's sort of uneven in its density. So it's probably just one random hit, but the original part of protein matches much better in this area. Okay, so talking about algorithms and applications, this application can help you with your algorithm if you take it further. Okay, so now we have two aligned regions, and you can keep studying this region by, so we found that there is, there is protein hit, but we want to take a look at the DNA hit. So we compare this sequence on the right, we run instant BLAST-n, and we compare it to the sequence on the left. So we display the result of BLAST-n again graphically, and we have it as just with one nucleotide precision. Again, we will notice that there is not much conservation in introns, and these red lines, they correspond to indels. So again, the indels are just get together inside introns, but not in exons, because nature will not tolerate them inside exons. Okay, so just a quick observation by just looking at the graphics. If you zoom further, you will see, yes, this is just identical sequences, DNA sequences with some mismatches here, or if you look at intron part, these are indels. You can align maps using different methods, and one of them is using orthologous gene pairs. You see this line, it connects the orthologs. So if you click a gene and there is a tab orthologs, which lists of all the orthologs of the given gene in other organisms. For example, this is rice, and we see that there is ortholog in sorghum, in wheat and corn. You can call them up here to this interface and just see this all together. This is one way of aligning the maps. So you can zoom out and you will see that the neighboring genes are also sort of aligned. And you keep zooming out and you will see this is the structure when you align entire chromosomes. So you will see some rearrangements, translocations and inversions. For example, here you can click invert plate and you will just sort of untie this knot around this, so you can study it when everything is collinear. And then we'll put it back like this. And okay, so you can add other genomes here to the picture, and then we'll study the evolution and the just the history of rearrangements. I wanted to show also, beside the plant genomes, I wanted to show some data for human genome or viruses. And here we have a 45 gigabyte BAM file. So it's just whole genome sequencing for one of our developers, by the way. So I 45 gigabyte, and then it requires also index file here. And okay, the index is uploaded, and then it finds the match, so it asks on which genome you want to show this BAM track. If you select wrong version, it will find that something doesn't fit. Okay, so we select the old one, and the map names are matching automatically, and then we select next, we give it a proper color, and this is it. So now you can just work with this file, and so this is the chromosome data from the database with few tracks, and this is our file 45 gigabyte. We were training it also with 200 gigabyte file. It works fine so far. So we have also ClinVar data, so clinical consequences of known mutations. You just click and read all about that. You can also drag and drop marker files with other genotyping results and compare them to known mutations just to find out if the mutation is harmful or not. And this BAM tracks, this one was loaded before from URL, so it stays there. If it's a file, huge file that you just uploaded, like I did now, is a local file, then you will have to re-upload it again because it's too large, it doesn't fit the disk quota. But if you just point it as a URL, then it will stay there. Okay, so this is just a look at nucleotides there, or you can hide non-variants as we did before. Okay. What's also interesting is the data for human also contain a track, this file P100, which is a conservation track. So the score of conservation. They analyzed 100 species and just recorded the score, the conservation. So the higher, the more conserved region, and you see this. Of course, it's consistent with the exons. So the exons are conserved. Everything between them is free to fluctuate. Okay. So, and this is the data on the level of one value per nucleotide, and you see there's 250 million nucleotides in one chromosome, and it still shows every single one of them. I also wanted to show the functionality of Persephone with virus data. So we have some of them loaded, and the most popular, of course, the ones that that interesting these days, the bat coronavirus, also the one that comes from Wuhan, and the one that recent Delta from Bangladesh. Okay, so there are three sequences. we can just compare them using this instant blast feature. You just click and the sequences are compared. So every line here is a mismatch. So when you compare Wuhan and the later version of Delta variant, you can see that roughly there are 40 mismatches, which is consistent with the speed of like one mutation per 2 weeks. And of course, we're interested also in the spike protein. This one is spike protein, S protein. The red one means there is an insertion or deletion. So when we compare Delta variant to Wuhan, and we know this one is more transmissible, this could be attributed to these differences. So we miss a few nucleotides that result in amino acid changes. Okay, and also we notice that when we compare Wuhan version to the bat virus, there is also some mutation, insertion, deletion. It is somewhere here. Yes, here. This is the notorious furin site. You see this PRRARS VAS? This is the one. Okay, so when you compare the old one, it didn't have it. But what's interesting is this sequence. I was wondering what's going on here, where did it come from, and I just compared this sequence plus something else to human, and what I found is this this insertion matches perfectly. So insertion plus 2 flanking nucleotides, so 16 nucleotides GC rich. It matches in antisense to exon of human B-link gene, right? B cell linker, which is responsible for immunity, and there are known mutations. You see from CleanVar, if you have mutation in this gene, you will have problem with the viral infections here and there. So I don't know, I'm not a specialist in this. If anybody knows this stuff, my theory was that the RNA from this gene, mRNA, will be antisense matched to the mRNA of this gene, and could it silence it? Or the question is who is silencing who, but I think the number of copies of mRNA from virus is higher than this one for human. I don't know, but it looks interesting. I will be happy to hear some comments. Thank you.